Imagine, for just a second, trying to stuff 2 entire meters of sewing thread into a microscopic sphere, like something smaller than the head of a pin.
Right, which already sounds impossible.
Exactly. But now, imagine that you need very specific spots on that thread spots that are separated by, I don't know, hundreds of thousands of units to physically touch each other at the exact same millisecond.
Just to turn on a single light switch.
Yeah, just to turn on a switch. I mean, this isn't a magic trick, right? This is the actual 3-dimensional origami happening inside your cells right now. It's the physical folding of your DNA.
And, well, when that folding goes rogue, the light switches that get flipped on can cause some incredibly aggressive diseases, like mantle cell lymphoma.
Which is why we're doing this deep dive today. We are jumping into a really dense, just completely fascinating May 2025 paper from Nature Cell Biology.
Yeah, it's a phenomenal piece of research.
Our mission here is to figure out exactly how these cancer cells manipulate the physical architecture of your DNA to survive, and how scientists are finally catching them in the act.
I mean, it's a remarkable paper because it forces us to shift our entire perspective. For decades, we've dealt with this almost overwhelming information overload in genomics.
Right, just looking at the sequence.
Exactly. We focused just on reading the literal letters of the DNA to see, what genes were mutated. But knowing the sequence just isn't enough anymore.
It's like having an instruction manual, but you can't just read the words, right? You have to look at how the pages are physically folded together.
That's a great way to put it. This research brilliantly exposes how specific proteins act as, like spatial traffic cops to shape the genome, and how cancer completely exploits them.
So I want to set the stage here, because to understand how a cancer cell hijacks this folding, we have to look at the sheer scale of the DNA thread. Let's talk about the cast of characters.
Sure, let's break down the terminology.
Okay, so you've got the promoter, which is essentially the engine of a gene. It's the starting line where the machinery attaches to read it. But the ignition switch, the remote control that actually turns that engine on, is called an enhancer.
And what's wild about the enhancer is the distance.
Right. This remote control can be sitting like 500,000 base pairs away from the engine on the linear thread.
And this is where the folding becomes so critical, because to push the buttons on that distant remote control, you need specific proteins. We call them transcription factors.
The fingers pushing the buttons?
Exactly. And the main star of this research is a lineage-determining transcription factor called EBF1. Its normal day job, basically, is to give B cells, which are a vital part of your immune system, their specific identity.
So it binds to the DNA and tells a generic cell, hey, you are going to be a B cell, and here are the genes you have to turn on.
Spot on. But, the thread doesn't fold itself. It needs a motor to actually move things around.
Yeah, and when I was looking into that motor cohesin, the best visual I could think of was a tiny zipline motor attached to a looped shoelace.
Oh, like that.
Right, like the motor stays in one place and just continuously pulls the string of the shoelace through itself, extruding the DNA to make the loop bigger and bigger.
That zipline visual is perfect. Cohesin is constantly extruding the DNA, pulling it through this molecular ring, and that creates these massively dynamic loops.
Okay, so we have the engine, the distant remote control, the transcription factor pushing the buttons, and the motor pulling the loops. Which brings us to the crime scene.
Mantle cell lymphoma.
Yeah, MCL, an aggressive B-cell cancer. The researchers found that this cancer is entirely addicted to that EBF1 protein. If you take EBF1 away, the cancer cells just die.
Which presents a huge problem for scientists.
Right, because reading the methodology, I hit a logical wall. Normally, if scientists want to see what a protein does, they use CRISPR to knock out the gene, wait a few days, and look at the result.
Right, that's the standard playbook.
But if deleting EBF1 kills the cell, how on earth do they study the precise mechanics of the DNA folding without just looking at a dead, rotting cell?
That is the exact methodological trap that has frustrated researchers for years. I mean, if you use CRISPR, the cell slowly starves and dies over several days.
So the data is basically ruined.
Yeah, by the time you try to map the DNA architecture, you are looking at biological rubble. The immediate mechanism of what EBF1 was actually doing is totally lost.
So how did they fix it?
To solve this, they bypassed CRISPR entirely and used a brilliant piece of engineering called the D-tag-degradation system.
It's essentially a molecular kill switch.
A highly precise, real-time kill switch. They genetically modify the lymphoma cells by attaching a molecular self-destruct tag, the FKBP-Ki tag, directly to the cell's EBF1 proteins.
Okay, so the tag is on there, then what?
Then they simply introduce a specific chemical, the DTAG molecule, into the environment. When that chemical enters the cell, it flags the EBF1 protein, and the cell's own waste disposal machinery instantly shreds it.
Wow, and they were able to wipe out all EBF1 in just 2 hours.
Just 2 hours. The temporal resolution changes everything.
That's insane. 2 hours to destroy it. And they found that if they wash the chemical away, the EBF1 protein levels fully restore in just 6 hours.
Exactly. And they proved that doing this for 24 hours barely impacted the cell's overall health.
So they're capturing pure, unadulterated architectural mechanics in real time.
They isolated the variable perfectly. By flipping the switch, they found over 7,000 specific EBF1 binding sites across the genome that responded beautifully to this quick removal and restoration.
That is incredible. Okay, let's do a quick recap of this first part for everyone listening, and we'll do this throughout the deep dive to keep it all straight.
Good idea. There's a lot of terminology.
So, bullet summary. We defined promoters as the engine, enhancers as the remote control, EBF1 as the finger pushing the buttons, and cohesin as the zipline motor pulling the legs. We learned EBF1 is vital for mantle cell lymphoma, and we explored that awesome DTAG system, which is a 2-hour kill switch that lets us watch the DNA collapse in real time.
This covers the basics, yeah.
So, quick question to you, the listener. If you could attach a 2-hour biological kill switch to any single process in a cancer cell, just to watch how it falls apart in real time before the cell dies, what would you choose? It really makes you think about what we've been missing.
And, well, that level of precision is what led them to their next major hurdle.
Right, because we know EBF1 binds to those distant remote controls, the enhancers.
Yeah.
But how does it physically bring them all the way over to the promoter engine?
To actually visualize that happening in 3D space, standard tools were never going to work.
Yeah, because traditional genome sequencing, like Hi-C, is essentially a population-level tool. You have to crush up millions of cells to get an average reading.
It's a huge limitation.
If we think about this like trying to understand city traffic, traditional sequencing is like taking a blurry satellite photo of the entire city averaged out over a month. You might see a general red zone of congestion, but you can't tell me the exact path a specific car took on a Tuesday at 4 p.m.
And that average is exactly what hides the real biology. A blurry satellite photo doesn't cut it. The researchers needed an upgrade, so they utilized a technique called ORCA.
Which stands for optical reconstruction of chromatin architecture, right?
Yes. And going back to your traffic analogy, ORCA is like attaching highly precise 3D GPS trackers to 30 specific cars and tracking their exact coordinates in a single cell in real time.
The way they pulled this off is just staggering to me. They targeted a 900 kilobase region of DNA. That is a massive stretch of the genome.
And they didn't just look at it all at once. They used unique fluorescent barcodes to essentially paint the DNA.
Like glowing labels.
Basically, yeah. They labeled one specific 30 kilobase segment, took a 3D image, and then removed the label. Then they labeled the next segment down the line, imaged it, and removed it.
And they did this sequentially for 30 distinct segments.
Yes, across more than 100,000 individual single cells. It is sub-diffraction 3D GPS for the genome.
Wow. And they aimed this incredible camera right at a massive oncogene, a major driver of cancer called MYC.
Right, the MYC gene. It has its promoter engine, but it is controlled by two separate massive super-enhancers, which they labeled E1 and E2.
And the distances here are wild. E1 is sitting 525,000 base pairs away from the promoter. E2 is 433,000 base pairs away. So you have these two massive remote controls sitting 500,000 units down the thread, and EBF1 is binding out there. What did the ORCA camera actually see?
Well, it saw a structure that population average sequencing could never confidently prove. EBF1 doesn't just grab one enhancer and touch it to the promoter in a simple pair.
It does more than that.
It orchestrates what they call a multi-enhancer hub. It creates a complex 3D intersection where the first superenhancer, the second superenhancer, and the MYC promoter all converge at the exact same microscopic point in space.
A three-way biological intersection, and they could actually prove causality using that DTAG kill switch.
Exactly. When they dropped the chemical in and destroyed EBF1, ORCA showed this massive 3D hub completely unraveling. The enhancers just drifted away into space.
And when they washed the chemical out,
the hub rapidly refolded in a matter of hours, specifically prioritizing these three-way intersections to blast the MYC cancer gene back into overdrive.
The speed of that reassembly is terrifying. But there is a detail in the ORCA data that is even more profound, and I want to make sure we don't skip it. It goes back to why the population average is so misleading. They looked at single-allele dynamics.
Yes, this is a crucial finding.
Okay, let's unpack that, because an allele is just a copy of a gene. You inherit one copy from your mom, one copy from your dad. Both of those copies are sitting inside the exact same nucleus of the exact same cell. Are you telling me these two copies of the MYC gene are doing different things at the exact same time?
They are completely uncoordinated, which is mind-blowing. In these untreated cancer cells, the ORCA camera revealed that the three-way intersection only happened in about 20% of alleles at any given moment.
Wait, really? Only 20%?
Yeah. One copy of the gene might be tightly folded into this supercharged hub, blasting out cancer-causing instructions, while the other copy right next to it, inside the same nucleus, is completely unraveled and quiet.
That completely shatters how I used to think about a cell's behavior. If you take 1 million cells and run them through a blender for traditional sequencing, you just see a mild, permanent, static connection across the board.
Because it averages out that 20% across everything.
Right. You totally miss the reality that these hubs are highly dynamic, flashing on and off independently at the single allele level. It makes you realize how many drug targets we might have missed simply because our maps were too blurry.
The dynamic nature of it is the key to everything.
Okay, time for our second recap. So, bullet summary. We learned about ORCA, the subdiffraction 3D GPS for DNA. We examined the MYC gene and its two distant super-enhancers, and we discovered that EBF1 instructs multi-enhancer single-allele 3D hubs that flash on and off.
That's the core of the imaging data, yes.
So, question to you listening. Think about the difference between a population average and a single-allele tracking system. How might zooming into individual uncoordinated alleles change the way we design targeted cancer therapies? If the targets are flashing on and off, how do we hit them?
And that question brings us perfectly to the biggest mechanistic mystery of the paper. We know EBF1 is the critical player. We know it builds this MYC hub. But how does EBF1 physically pull these massive DNA strands together from 500,000 base pairs away?
This was my very first thought when reading the paper. If EBF1 is sitting on an enhancer that is hundreds of thousands of units away, does it act like a magnet? Does it physically grab the DNA thread, reach across the nucleus, and yank the promoter towards it?
It is a logical hypothesis, but the physics of the nucleus don't really support it. EBF1 isn't actively reaching out and pulling anything.
Oh, interesting. So what is it doing?
To explain the how, the researchers validated what is known as the permeable barrier model. And to understand it, we need to bring back your zipline motor analogy.
Okay, so we have cohesin, our little motor, or let's think of it as a fast-moving train that is constantly moving down the DNA track, pulling the thread through its ring and making the loop bigger.
Right. Now imagine EBF1 is not a magnet. Imagine EBF1 is a heavy speed bump, or a toll booth, placed directly on the DNA track at the exact location of the distant enhancer.
Oh, I see where this is going. The cohesin train is zooming along, pulling the DNA thread, expanding the loop, and suddenly it hits this EBF1 toll booth sitting on the enhancer.
Exactly. And because it hits that toll booth, the cohesin train stalls, it pauses. And because the motor stalls at that exact geometric location, the DNA begins to bunch up right there.
Like a molecular traffic jam.
Yes. This traffic jam effectively forces the distant enhancer, where the toll booth is physically located, and the promoter, which is being pulled into the loop by the motor, into close physical proximity. The hub forms simply because the motor got stuck at a specific checkpoint.
That is deeply elegant. It's not a magnet, it's just physical tension and traffic control.
Well.
But the model specifically calls it a permeable barrier, meaning it's a toll booth, not a concrete barricade.
That permeability is the most crucial part of the mechanism. The cohesin train stalls, it slows down, and it bunches up the DNA to form the hub. But occasionally, the motor manages to slip past the EBF1 protein and keeps moving down the track.
Oh, which perfectly explains why the single alleles were flashing on and off in the ORCA images.
You got it.
If EBF1 were a hard brick wall, the loop would just hit it and stay there permanently. But because it's a permeable speed bump, the cohesin stalls, the hub forms, then cohesin slips past, the hub dissolves, and then another cohesin train comes along and stalls again.
The 3D hub is constantly forming and dissolving. It connects the physical mechanism directly to the visual data we saw with ORCA.
That is fascinating. But of course, in science, proving this happens in one specific B-cell cancer isn't enough, right?
No, you have to ask, is this just a weird isolated quirk of mantle cell lymphoma? or as it's a fundamental rule of biology.
And this is where the paper delivers its final masterstroke. They wanted to see if this permeable barrier model held up elsewhere, so they completely switched disease models.
Yeah, they looked at TAAL, which is T-cell acute lymphoblastic leukemia. It's a completely different cell type and a completely different cancer.
And the dynamics changed entirely, didn't they?
Completely. In TAL, that EBF1 protein isn't even expressed. It's not the main player at all. Instead, a completely different lineage-determining factor called TCF1 acts as the toll booth.
And TCF1 doesn't bind to the same enhancers.
No, it builds a hub using two completely different MYC enhancers, which the researchers call TE1 and TE2.
And just to highlight the scale again, TE1 and TE2 are located a staggering 1.5 million and 1.85 million base pairs away from the promoter.
It's a massive distance.
1.8 million units away, and TCF1 acts as the exact same kind of speed bump to cause a traffic jam and fold them all together.
Yes, the mechanism is identical.
Yeah.
But then the researchers looked at something truly terrifying about how cancer adapts. They examined TAL cells that had become resistant to chemotherapy drugs.
Okay, drug-resistant cancer, what happens?
In these drug-resistant cells, the cancer physically loses access to the TE1 enhancer. It's just gone.
So without that enhancer, the toll booth is gone, the hub should collapse, the MYC gene should turn off, and the cancer should die.
Exactly what you'd expect. But instead, the cancer pulls a vanishing act. To survive the drugs, the T-cell cancer actually turns on EBF1, the protein normally restricted to B-cell cancers.
Wait, it wakes up a B-cell protein inside a T-cell cancer?
Yes. It activates this foreign protein, and EBF1 repositions itself onto the remaining TE2 enhancer to hijack the toll booth mechanism.
That is unbelievable. The cancer realized its toll booth operator was gone, so it reached into a completely different genetic toolkit, woke up the B-cell protein, and swapped out the operator just to keep the traffic jam going and survive the chemotherapy.
It adaptively reorganized its own 3D architecture to stay alive. This proves an overarching fundamental biological rule.
Which is?
Lineage-determining transcription factors act as permeable barriers to loop-extruding cohesin. They radially position enhancers and promoters together to drive gene expression. Whether it's EBF1 or TCF1, the physical physics remain the same.
Wow. Okay, our final quick recap, bullet summary. We explained the permeable barrier model. EBF1 acts as a speed bump, not a magnet, to stall cohesin trains. This forces 3D clustering, and we saw this identical mechanism validated in T-cell leukemia with TCF1.
And we saw how cancer can swap the toll booths to become drug-resistant.
Right. So my final question to you listening for this section, if cancer relies on these highly specific toll booths to fold its DNA, could we design a new class of drugs that simply flattens the toll booths, letting cohesin pass by without turning on the cancer gene?
It really reframes how you look at the resilience of these diseases.
It really does. We started this deep dive with an almost impossible visualization stuffing meters of thread into a microscopic sphere. And piece by piece, this paper decoded the origami.
We covered a lot of ground today.
We watched molecular trains called cohesin ride along the thread. We saw speed bumps like EBF1 and TCF1 cause precise traffic jams. We saw cancer swap out those speed bumps to survive drugs. And we visualized it all using the high-tech 3D GPS of ORCA.
These researchers successfully deconstructed the physical mechanics of survival. And this is exactly why this research matters beyond just one type of lymphoma. We are officially moving past the era of simply reading the linear genetic sequence.
It's a whole new frontier.
The future of biology and the future of life-saving medicine is spatial. It is not just about the words written in your DNA instruction manual. It is entirely about how those pages are physically folded together. Once we understand the rules of the folding, we can finally start to unfold the disease.
Which brings us to the ultimate question for where we go next. We've seen that nature relies on these highly specific permeable molecular toll booths to fold its DNA. So what is stopping us from engineering our own?
That's the million-dollar question.
Right. Could we one day design a synthetic protein, inject it into a patient, and have it park itself permanently on the genome? Imagine an unpermeable brick wall that completely blocks the cohesin train, effectively trapping cancer genes in an off position forever.
That would be the ultimate cure.
Think about that the next time you hear about gene editing. The future of medicine isn't just about cutting the DNA thread, it's about controlling the traffic.